Hi, I'm Maria Inés Freiberger. First, I want to say thanks to the organization for choosing our investigation to be present at this Congress. This is a collaborative research with the Laboratory of Protein Dynamics in the University of Debrecen in Hungary, and with the Center of Theoretical Biological Physicists in the Rice University in Houston. The title is Frustration Leads to Fuzzy Interaction in Disordered Protein. First, I'm going to talk about fuzzy regions. Disordered protein often display different structure when bound with different partners. This is termed as a fuzzy binding. These binding modes range from nearly fully ordered to rather disordered state in the bound complex. The structures can change with post-translational modification or in varying cellular conditions. Fuzzy binding enables disordered protein to interact not with every biomolecule but specifically with a defined multiplicity of partners. The structural change of disordered protein upon binding ranging from decreases to increases disorder. The spectrum of this binding mode can be quantified using conformational entropy. From disorder to order region, where conformational entropy is reduced. Disorder to disorder regions where conformational entropy remains high or increased upon binding. Binding mode of context-dependent region may also depend on the partners and cellular environment. In the figure, we can observe the binding mode of the disordered protein sample a wide spectrum of structure and dynamics. Here we can observe an example for each binding mode. Disorder to order region gain a well-defined conformation upon binding. Disorder to disorder region remain disordered while interacting with their partners. Context-dependent region exhibit both type of behavior, depending on the partner or experimental condition. Although the biological significance of fuzziness has been established, understanding how diversity and specificity are reconciled required to use the energy landscape theory. A protein does not fall by testing random interaction, but it does so by minimizing its energy conflict, which is known as the minimal frustration principle. However, this principle does not imply that there cannot be certain conflict remaining in the native state. Each time a native interaction is established in the polypeptide chain, the energy goes down more than it would in the case of forming a random interactions. As a consequence of this cooperativity between native interaction, the global form of the energetic landscape of natural protein has the form of a corrugated funnel where there is a strong energy bias toward the native state. Now, the question is, this conflict remaining in the native state are important for protein function? Can we quantify this conflict? The frustratometer is an algorithm based on the energetic landscape theory. First, it defines the contacts in the structure. Next, calculate the native energy for each interaction, perturb the native interaction creating decoy, and measure the energy. Then, calculate the energy distribution for the decoys and compare the native energy to the overall distribution. To compute the frustration index for interacting pair of amino acid A and G, simultaneous mutation on the residues A and G are made. There are two ways for localizing frustration at the pairwise contact level. These ways differ in how the set of decoy is constructed. In one choice, the decoy set is made by randomizing only the identity of the interacting amino acid AG, keeping all other interaction parameters at their native value, called mutational frustration. A second way to quantify pairwise local frustration, the residues, are not only changed in identity, but also can be displaced in location, called configurational frustration. The frustration index are classified into three classes. If it's less than -1, the contact is highly frustrated. So, these contacts are in conflict with the structure. If it's greater than 1, the contact is minimally frustrated. This contact is good for the stability of the structure, and if it's between -1 and 1, the contact is neutral. In this study, we have performed a systematic analysis of frustration on complex of 138 disordered proteins. This protein contains disordered region in the free form, while exhibit different binding mode in the bound form. For this analysis, we use DR and CDR structure because DDR does not have the fuzzy residue crystallized. DR structure are PDB with resolution higher than 3 angstrom, with missing electron density for at least 5 residues. We then collect all available bound state structure and filter those where the disorder region was observed in all the complexes. We analyze the interface residues and selected those regions where at least one residue mediated intramolecular interaction. The DR dataset containing 97 non-redundant disorder regions. For CDR structure disorder regions, which were observed as both folded and disordered in their complex structure, depending on the partner, were defined as context-dependent region. We assembled context-dependent region with a minimum length of 5 residues in the CDR dataset. This dataset contains 93 non-redundant disorder regions. We use a part distribution function to measure the local frustration around the fuzzy region between the alpha carbon of the protein and those residues that are fuzzy. In x-axis are represented the distance in angstrom, and in y-axis the value of the GR. GR value were normalized such that the value of the function at 20 angstrom is equal to 1. For both fuzzy region, DR and CDR, we use a non-redundant dataset. As a result, first I'm going to show the result for DR regions. We can observe an example of a frustatogram of a protein structure, and in yellow are colored the fuzzy regions. In red lines are the highly frustrated contact, and in green are the minimal frustrated contact. Also, we can observe a part distribution function, GR, for configurational frustration index. In black line are shown all contacts. In green, minimally frustrated contact. In red, highly frustrated contact, and neutral are gray. In the GR of the left, we observe a frustration pattern for fuzzy region, and we see an enrichment of highly frustrated contact around 2 and 4 angstrom. The other GR, as a control, we choose random residues that are not fuzzy. We also can observe an enrichment of highly frustrated contact, but are slightly less frustrated than those of disordered regions. For CDR region, we can observe also an example of frustatogram of a protein structure. Here we can observe in the GR of the left the frustration pattern for fuzzy region, and also we see an enrichment of highly frustrated contact. But in this case, we have 2 peaks, one around 1 angstrom and the other around 2 and 4 angstrom. And the other GR, this is a control. We also can observe an enrichment of highly frustrated contact, but are slightly less frustrated than those of disordered regions. Here we can observe 2 histograms for fuzzy residues that are involved in binding, showing pink, and non-binding contacts showing blue. And in the x-axis are represented their configurational frustration index. The above histogram is for DR and the other below is for CDR. We observe that those residues which do not form contact with the partner exhibit higher frustration index than those which directly form intermolecular contact. In this case, we analyze fuzzy residues that are inter- and intramolecular. We can observe a GR for both types of residues. The above GR is for DR and the below for CDR. To the left are shown the intermolecular residues, and to the right the intramolecular residues. We observe an enrichment of highly frustrated contact in both, but intramolecular residues exhibit higher frustration index. Here we analyze monomers that are ordered in the free form, but get fuzzy in the bound form. We analyzed 52 non-redundant fuzzy regions, and in the figure we can observe an example of frustatogram, and in yellow are colored the fuzzy region, and the year for configurational index on the left, and on the right for mutational index. We can see two peaks of an enrichment of highly frustrated contact in both frustration index around the fuzzy regions. These results are consistent with the idea that the folding of disordered region upon their target does not result optimal structure in their complexes. What is the molecular basis of target selection? To answer this question, we analyze complexes of the same disordered region interacting with different binding partners. For this example, we consider the fuzzy region of the tyrosine protein kinase. In this protein, the number of the residues that are fuzzy are the 545 to 556. The protein PDB-ID 1K2P are unbound and has a homodimeric structure. Protein PDB-ID 5J87 are bound with N42 and has a homotetrameric structure. Figure shows both the structure and the density of local frustration calculation for configurational frustration index of this protein. In line are shown 1K2P and in dotted line are shown 5J87. We observed that the density of contact around each structure have different frustration pattern for the fuzzy region. Also, we see 5J87 is more frustrated than 1K2P. Here we observed other example. We consider the fuzzy region of nitrogen regulatory protein. In this protein, the number of the residues that are fuzzy are 38 to 53. For the protein structure with PDB-ID 3MHY are bound with multiple of ligand and has a homotrimeric structure. PDB-ID 4CNZ are bound with ADP and has a homotrimeric structure. Figures show both the structure and the density of local frustration calculation for configurational frustration index of this protein. In line are shown 4Z and Z, and the dotted line are shown 3MHY. We observed that the contact density around each residue show difference in frustration pattern in the fuzzy region, and 4C and Z are more frustrated than 3MHY. As a conclusion, here we performed a systematic analysis of complexes of disordered protein and demonstrate that even after binding, their folding is far from being optimal, consistent with early results on individual cases. We find that both the disordered and structured region are enriched or in highly frustrated interaction in the bound state of disordered proteins. Interface contacts decrease the frustration in the disordered protein as compared to the free state, but the interaction often still remains suboptimal. Frustration is higher in fuzzy regions, which are observed to have multiple binding modes. We demonstrate that the fuzzy region displays distinct frustration patterns with different partners. Rationalizing one-way limited specificity and versatility can be encoded because structure is not fully optimized. This is my work team, Professor Diego Ferreiro from Protein Physiology Lab in Universidad de Buenos Aires. He is my advisor, Professor Peter Wollines from the Center of Theoretical Biological Physicists in Rice University in Houston, United States. The PhD student, Victor Ambrus from Debrecen University in Hungary, and Professor Monika Fuchsreiter from Debrecen University in Hungary.